Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my lecture. So today we're talking about respiration in plants. So we'll try to finish off respiration in plants in today's lecture. So I'm just going to give you a recap so that I want to finish the portions very soon so that you can have self-study or you can have time for your self-study. Hello, yes Shalini, a very happy good evening. So thank you so much for joining my lecture. I'm really sorry for all those who came at 5 and found the lecture to be 5:15. So that was due to an issue at the back end. So sorry for the apology. So this is only for today. So we have the lecture at 5:15. So from tomorrow it will be at 5 as usual. Okay. So today we'll finish off respiration in plants because respiration in plants is a very small chapter. So where you have to talk in detail about glycolysis, your Krebs cycle and your ETS. Apart from that, it's a very easy chapter which we can finish it off in one lecture. So I'm just going to give you a recap of this. Hi Ved, yes. Hello Antonia B, a very happy good evening. Yes, I'm doing good. How are you? Thank you so much for joining my lecture today. So please don't forget to like, share and subscribe. And I have another important announcement to make to all of you. So today we are in respiration in plants, right? So I will finish up respiration today in class. Hi Tamar Saran, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. The announcement that I want to make, so tomorrow we are going to talk about your strategies and your microorganisms, your strategies for enhanced food production and your microbes in human welfare. So both are two simple chapters, so I'm not going to revise them theoretically. So instead I will become 20 questions from each of the chapter and finish off the discussion. I'll make sure I will finish off the chapter in such a way that all the topics of the chapter are covered in that question, so that it is easy for you to revise, and we will not extend 2 more lectures for strategies and 1 for microbes. Both are easy, right? So for these 2 chapters I will bring questions directly and discuss. And on Saturday, so today if respiration is done, in your plant physiology we have 3 more chapters, right? So one is mineral nutrition, transport in plants, and then we have your plant growth and development. So all these 3 chapters also I will discuss in the form of questions. So yes, you will have a recap. So 20 questions from each chapter. So we will be done with this chapter. So next week we will be just left with biotechnology, which I will finish. All right? Yes. Yes, Shalini, hello. Yes, I'm good. How are you? So I'm good. Thank you so much and thank you for joining my lecture. So Balakrishna Reddy, yes, a very happy good evening. So thank you guys for joining my session well on time. So please don't forget to like, share and subscribe. So let your friends also know here ma'am is teaching the entire syllabus, and very soon we are going to revise. We are going to have a full portion mentee. So we will have a motivational session. We have one more interesting session for you. So please don't forget to share the session with your friends. So more number of participants, more people can benefit, right? So thank you so much, and here we move into respiration in plants. So this is a beautiful chapter, yet alone if you understand the concept. So let's finish it off in one lecture. So let's be a bit fast. If you do not understand, stop me then and there. I'm ready to explain once again. Fine? Yes. So we are going to talk about, do plants breathe? So plants do not have alveoli, so plants do not have lungs. So how are they going to breathe? So is there a specific mechanism by which plants are going to breathe? So what is glycolysis? What is fermentation? What is aerobic respiration? Then we have what is amphibolic pathway, and at the end we are going to talk about what is respiratory quotient. So these are some of the topics that we are going to discuss today, and we are going to finish off this chapter of plant respiration. So moving further, how do we define respiration? So photosynthesis is the process by which plants prepare food, and photosynthesis takes place within the chloroplast. So chloroplast is a double-walled organelle that is present in your plant cells. The breakdown of complex molecules to eat energy in the cytoplasm and in mitochondria is your respiration. So photosynthesis gives you glucose, right? Now breakdown of this glucose to give you energy is the process of respiration which happens in all the plant. So the breaking of the CC bonds of the complex compounds, your glucose also has CC bonds, right? So the breaking of CC bonds of complex compounds, the oxidation within the cells leading to the release of the considerable amount of energy is known as respiration. In photosynthesis, plants make up carbon dioxide. In respiration, plants use this carbon dioxide to break down and form energy for the day-to-day activities. Fine? Yes? Yes, Swami. Hello, hi. A very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So compounds that are oxidized during this process are known as your respiratory substance. So if I say photosynthesis produces glucose, so here your glucose is being broken down, right? So glucose is the substrate. It is known as the respiratory substrate. So you can have carbohydrates, proteins, fats, and even organic acids which can be used as your respiratory substrate. So energy is not released in a single step, but it is released in a series of steps so that energy is not lost. Ma'am, is this energy released in a single step? When I say if your carbohydrates are broken down, is it happening in a single step? So I understand nothing happens overnight, right? So similarly, this breakdown also does not happen in a single step. It happens in multiple steps, so that energy is conserved here. So when your energy is coming, I have to store the energy, right? It should not be in such a force that all the energy is being lost. So it has to happen in multiple steps, right? So this is the first point in your respiration. So you should understand what is respiration, okay? So Sandhya, yes, hello, very happy good evening. So are you new to my session? So thank you for joining my session. So please do like, share and subscribe, and thank you once again for joining my session. Fine. So now, how do plants breathe? So first thing how plants breathe is plants help in the breakdown of certain products, especially your glycolysis. So we have two types of organisms, aerobic and anaerobic. Irrespective of whether they are aerobic or anaerobic, they all follow the first method, that is glycolysis. So the first method that is there in all plants is your glycolysis. So glycolysis happens in all the plants. It happens in both your aerobic as well as your anaerobic plants. So this is something common. So where does your glycolysis happen? So glycolysis happens in your cytoplasm. So glycolysis is also known as your EMP pathway, Embder-Mayen-Paranoff pathway, because these are the three scientists who discovered this pathway. They found out that yes, a process is happening like this. What is the starting point of this glycolysis? So here your substrate is glucose. So glucose is broken down in a 10-step reaction to your pyruvic acid. So this is your substrate and this is your product. So you have one molecule of glucose which forms two molecules of pyruvic acid. So this is the nutshell of your glycolysis, right? And this is an enzyme catalyzed reaction. Hi, yes, welcome to my session. A very happy good evening. So thank you for joining my session. So please do like, share and subscribe. Fine, so this is one part of glycolysis. So this happens in both your anaerobic as well as your aerobic organism. So glycolysis, so here is the structure of glycolysis. If you see, it is a 10-step reaction. So this is how it happens. So you have glucose. So first glucose is getting converted into glucose 6-phosphate. So this glucose 6-phosphate from glucose is converted by an enzyme known as hexokinase. So you have to remember at least the first enzyme. So wherever you have kinase, the function of kinase is to add a phosphate group. So this you have to discuss, okay? Yes? Okay, so today you will be present for the whole lecture. Okay, so thank you. That's really nice. Fine. So from glucose to fructose 6-phosphate, you have your isomerase. So you have your okay. So this is isomerase because glucose and fructose are the same 6-carbon molecule, right? And here one more phosphate is being added. So fructose to fructose 1,6-phosphate. So here the enzyme will be your phosphofructokinase. So remember only this much, so that is more than enough. So what are the things that you will remember? So glucose being broken down into pyruvic acid is a 10-step reaction. So first glucose is converted into glucose 6-phosphate, right? So this glucose 6-phosphate from glucose is done by your hexokinase. And here you have a utilization of one ATP molecule. So then you will understand from glucose it is converted into fructose 6-phosphate. So here the enzyme is isomerase. So fructose to fructose 1,6-bisphosphate. So this is your phosphofructokinase. So remember this and then the later goes on because these three enzymes are important. Okay? Yes? Yes, you're right, Wade, you're right. Okay, fine. So now you have to remember, you don't have to remember all the 10 steps, that's completely fine, but remember it is 10-step process. So here you have to remember the net ATP and the NADPH count. So what is the net ATP and the NADPH count? So for the process of glycolysis, so you have 2 ATPs that is being used. So 2 ATP is used and you have 2 ATP, so you have 4 ATP being produced. So in your glycolysis process, you have 2 ATP used and you have 4 ATP produced, okay. So thus the net is your 2 ATP. So the net result is your 2 ATP and we also have 2 NADH. Right, so these are the high energy bonds. So we have ATP and NADH because that is the purpose of having respiration. Okay, done. So convert NADH in terms of ATP. So 1 NADH is equal to 3 ATP. Fine. So now here do, so it is 2 ATP plus 2 into 3, 6 ATP, right? I am converting NADH in the form of ATP, right? So now moving forward, we have 8 ATP in your glycolysis. No, Anthony, don't confuse much. You don't have to understand the full cycle, okay? So remember fructose 6-phosphate. So from fructose 6-phosphate, it becomes triose phosphate. So this can be converted into DHAP as well, and then the final product is pyruvic acid. You don't have to remember the entire cycle, so don't confuse, okay. So this is the turnover in your glycolysis. Now moving to the next step. So what can the pyruvic acid have a fate? The pyruvic acid can have 2 fates. So here the end product is your pyruvic acid, right? So your pyruvic acid can have 3 fates. So what are the 3 fates of your pyruvic acid? So your pyruvic acid can either undergo fermentation reaction. So it undergoes fermentation reaction in case of anaerobic organisms. So we have 2 types of fermentation here. So we have net, okay. So actually the number of ATP produced is 4 and number of ATP that is utilized is 2. So 4 is being produced and 2 is being utilized. So the net ATP is 2. So it is 2 ATP plus 2 NADH, but here we have converted NADH also in terms of ATP. So it is 8 ATP. Okay, so if according to the question you will make up your answer. If the options have 2 ATP and NADH, you will go for that. If the options have only 2, 4, 8, so you will write 8 ATP. Okay, yes, fine. So your pyruvic acid undergoes fermentation in your anaerobic organism. So this can be alcoholic fermentation. So it releases alcohol plus carbon dioxide, or it can be your lactic acid fermentation. So lactic acid fermentation is in case of your muscles and in your lactic acid bacteria. Fine. Okay. So this is fermentation, okay. The another fate of your pyruvic acid is that it can enter into aerobic respiration. So in aerobic respiration, you have your TCA cycle, you have your electron transport system. So these are the 2 fates of your pyruvic acid. So one is lactic acid fermentation, right? The other is alcoholic fermentation, or it is your aerobic respiration. So in fermentation, we have anaerobic conditions in many prokaryotes and unicellular eukaryotes. So complete oxidation of glucose to carbon dioxide and water happens in the Krebs cycle. This is aerobic respiration. So fermentation happens in anaerobic condition and aerobic respiration happens in aerobic organisms. Fine? Okay, so moving further, so this is how it is. So glucose is broken down into steps like this, and finally you have pyruvic acid. So either it can break down to form ethanol plus carbon dioxide in case of your yeast, or it can break down to lactic acid plus carbon dioxide in case of your lactic acid bacteria or your muscles. Right? So this is the anaerobic part. Now we are going to talk in detail about the aerobic part of the respiration. So aerobic part occurs in mitochondria. Right. So the final part of your glycolysis, that is your pyruvic acid, it quickly moves into mitochondria from the cytoplasm for the process of your aerobic respiration. So what is the crucial events here? So first you have the stepwise removal. So everything happens, okay. So this happens in the matrix. So first you have what is known as the link reaction. So the link reaction also happens in the matrix of the mitochondria, and the electron transport system happens in the inner membrane of the mitochondria. So we will talk about it. So actually understand your aerobic respiration occurs in mitochondria. So your pyruvate, which is the byproduct of your glycolysis, that enters into the mitochondria from the cytoplasm. Okay? Yes? So lactic acid in aerobic, lactic acid in your lactic acid bacteria and in your muscles. Your muscles also produce, right? So aerobic respiration is what happens is glycolysis, TCA cycle and your electron transport chain. Okay, Antonia B? Yes? Fine. So the pyruvate that has moved from the cytoplasm into the mitochondria, it breaks down, it undergoes a decarboxylation reaction. When I say decarboxylation reaction, one carbon dioxide is removed, right? And then it forms an acetyl-CoA group. So now this acetyl-CoA group moves into the TCA cycle or the tricarboxylic acid cycle. So this is known as a link reaction. So what am I saying? So we have your glycolysis. So glycolysis happened in your cytoplasm. So what happened in glycolysis? So we had glucose. So glucose was broken down into your pyruvic acid or your pyruvate. So one molecule of glucose, it gave birth to two molecules of pyruvate or pyruvic acid, right? So now this pyruvate or pyruvic acid, it moves into the mitochondria. Okay? So this is in aerobic respiration. I am talking about the aerobic respiration part. So now this pyruvic acid which moves into the mitochondria, it undergoes a reaction known as link reaction. Okay. So one molecule of pyruvic acid undergoes decarboxylation here. So decarboxylation means removal of carbon dioxide. So thus it forms an acetyl-CoA. So one pyruvate molecule forms one acetyl-CoA, and here the enzyme is pyruvate dehydrogenase. Pyruvate decarboxylase. Okay? Yes? And here you have the production of one NADPH. So for one molecule of pyruvate, you have one molecule of NADPH produced in your link reaction. Fine? Yes, 𝐓𝐤𝐿, yes, a very, very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Okay? So in your glycolysis, what was that thing? So you have 2 ATP plus you have 2 NADH. Yes? So here for one molecule of pyruvate, so I have 1 NADH, but since I have 2 pyruvate molecules, I have 2 NADH. Okay? Yes? Your teaching is crystal clear. Wow. So thank you. So which means you're not new. You have already attended my lecture. So thank you. Fine. So now this acetyl-CoA enters into the next one, that is your Krebs cycle. So Krebs cycle is also known as your citric acid cycle, or it is also known as your tricarboxylic acid cycle. So it is known as Krebs cycle because the name of the person who discovered the cycle is Hans Krebs. It is known as your citric acid cycle because the first stable product formed is your citric acid here. So your acetyl-CoA, it enters the TCA cycle and it forms the first stable compound, that is citric acid. Finally, through a series of steps, it forms out acetyl-CoA plus your carbon dioxide. So this is the end product of your citric acid cycle. Fine. So in your citric acid cycle, what is the turnover? Okay. Yes. So in link reaction, for one pyruvate molecule, 1 NADPH. So since I have 2 pyruvate molecule, I have 2 NADPH. Okay, Anthony. Hi, Naveen. Yes, hello. So, thank you so much for joining my session. So, please do like, share and subscribe. Fine? Yes. So, here I have 2 molecules of acetyl-CoA because I have 2 molecules of pyruvate. So, here I am talking for 1 molecule of acetyl-CoA. So, for 1 molecule of acetyl-CoA in your Krebs cycle, so what is the turnover rate? So, for 1 molecule, I have 2 NAD. I have 4 NADH. So, no, I have only 2 NADH. I have 2 NADH, DPH. So, I have 1 ATP molecule and I have 1 FADH molecule. Okay? Yes? So, now for 2 molecules of acetyl-CoA, so what is my turnover? So, this into 2. So, it will be 4 NADH, 2 ATP, 2 FAD. Right? So, this is at the end of your Krebs cycle. Fine. So, this is your tricarboxylic cycle. So, the first stable product in your citric acid cycle is your citric acid. So, hence the name. It occurs in the matrix of the mitochondrium. So, your Krebs cycle is your amphibolic pathway because it has both catabolic as well as anabolic reactions. So, all intermediates that are formed in this reaction are used in your synthetic pathway. So, here the substrate is acetyl-CoA, right? Fine. So, this is your tricarboxylic acid cycle which happens in the matrix of mitochondria. So, what do I mean by matrix of mitochondria? So, matrix of mitochondria is the cytoplasm of the mitochondria. Fine. So, all enzymes for this citric acid cycle are also present in the matrix except one. So, you have one enzyme, that is your succinate dehydrogenase. So, this succinate dehydrogenase is present in the inner membrane of the mitochondria. So, this is the only exception. Fine. So, in your glycolysis, how many ATP? We have 2 ATP and we have 2 NADH. So, in your link reaction for 2 molecules of pyruvate, we have 2 molecules of NADPH. So, moving forward to your TCA cycle or your Krebs cycle, for 2 molecules of acetyl-CoA, we have 4 NADPH. So, we have 2 ATP and we have 2 FADPH. Okay, so this is the net turnover. So, you can add everything and find out. So, totally I have 4 ATP plus 8 NADPH plus 2 FADPH. So, 1 NADH is equal to 3 ATP, right? So, 4 plus 24, so 8 3s are 24, right? And 1 FADH is equal to 2 ATP. So, plus 4. So, how much do I have totally? Yes. So, I have 32 ATP molecules. Clear? Yes. Any doubt? So, is this clear to everyone? Yes? No doubt? Okay. So, the last part of your electron transport or your aerobic respiration, it is ETS, right? So, finally, you should have ATP molecules produced or you should have your oxygen being utilized. So, the end product of aerobic respiration should be carbon dioxide plus water plus energy. So, carbon dioxide is formed in your TCA cycle. So, energy is being formed, right? But your water is not yet formed. So, your water will be formed in your electron transport system. So, after your TCA cycle, so first you have glycolysis, then you have link reaction, then you have your TCA cycle, now you have your electron transport system. Now that you have your high energy bonds, so we all accept the currency of INR, right? So, we have the Indian money. So, if someone gives you $2, you convert it into the Indian money and then you spend, because in India, so our currency is totally valid, right? Yes. So you go back and read your NCERT, it will be perfectly clear, okay? Yes. So we have to change it in our currency. So similarly, though we have ATP and NADPH, everything should be in the form of ATP. So how are we going to break this down? So now we have the NADPH and FADPH, which we are going to convert into ATP. So for that, we have the electron transport chain. Okay. So in the electron transport chain, the energy from the NADPH and the energy from the FADPH will be converted in the form of ATP, plus it will release water molecule. So this is the final product. Okay. So your electron transport chain, ETS, it happens in the inner mitochondrial membrane. So what is this inner mitochondrial membrane? Let me give you the structure here. So your mitochondria is a double membrane structure like this. Okay. So we have an outer membrane. So we have an inner membrane. So we have an intermembrane space. Right? Yes. So this is our matrix. So now at the end of your TCA, we have ATP, NADH and FADH, right? So ATP is the energy currency of the cell. We never say FADH or NADH as the energy currency of the cell. So now we are going to convert NADPH, FADPH in terms of ATP. So how it is going to be converted? So practically we say 1 NADPH is equal to 3 ATP. But in the body, it is converted in the process of electron transport system. Fine? Yes. So this electron transport system happens in the inner mitochondrial membrane. Okay. So this inner mitochondrial membrane is here. So how this is going to happen? So that we are going to see. So this is the net equation at the end of your Krebs cycle. So you have your ATP, you have your FADH and you have your 4 NADH. So this is for one molecule of your so this 4 NADH is for one molecule of pyruvic acid. So this includes your link reaction as well. So at the end you have 4 NADH, FADH and ATP. So this includes your 4 NADPH, which has your link reaction as well. Okay. So now we are going to talk about the okay. So fine, it is explained here. So you have 4 NADH, 1 FADH2, 1 ATP for one molecule of pyruvic acid form. So we have 2 molecules, so we multiply them into 2, right? So totally how much we have? So we'll have 8 NADH, so we have 2 FADH and we have 2 molecules of ATP. So don't confuse, this is just written, okay? You have one doubt. Ma'am, yeah, what is your doubt? What is your doubt? Yes, what is your doubt? Yes, Antonia B, what is your doubt? So you guys have a doubt in the ATP molecules here? Yes. So 2 ATP here, 2 NADPH here. So you have 2 ATP. So for one molecule of NADH, okay, so here it has to be 6. Actually, we have 6 NADPH for 2 molecules. If you see, you have 1 NADH, so 2 NADH and we have 3. So for one molecule, we have 3 NADH, right? So it is 6 here. So when I multiply 6 into 3, it will be 18. Yeah, so this is into 2 I have to do. So this is for, yeah, so this is for 2 molecules. So convert this into your ATP format. So this will be 18. So 18, 4 will be 22, 20. No, I am making a mistake. So this is your doubt, right? So till this it's satisfied. So in your Krebs cycle for one round we have 3 NADH. So we have 2 molecules of TCA. So now I am writing 6 here. So for one molecule in your TCA, It is 3 NADH, 1 ATP and 1 FAD. So for 2 molecules, this is it. Now I am converting everything in the form of ATP, right? So I have 4 ATP. So totally I have how many NADH? I have 10 NADH. Is that so? Yeah, so I have 10 NADPH plus I have 2 FADPH. Right? Yes. So now convert everything in terms of ATP. So 4 ATP. So this will be 30 ATP. So this will be 4 ATP. So totally we have 38 ATP or 36 ATP in your certain prokaryotes. So initially we got 32, right? So this was your confusion. Now clarified? Yes. So now we move forward with your electron transport chain. Yeah. So in your electron transport chain, what happens? So this is the structure of your electron transport chain. So directly we'll go to the figure, okay. So this is your electron transport chain. So this is the matrix of the mitochondria. This is the inner mitochondrial membrane and this is the intermembrane space. Okay, so whenever energy has to be produced, so you have to have a proton gradient. So that is what we spoke about in your chemi-osmotic hypothesis, right? So we have a H plus gradient. So this H plus gradient, when it breaks, we have the production of ATP. So when this breaks, it produces ATP. So we are going to have high H plus ion gradient in your intermembrane space. So this is going to have more H plus ions compared to your matrix region. Fine. So first you have your NADH that is produced from your Krebs cycle, glycolysis and the Glink reaction. So your NADH plus H plus is converted into NAD plus. This is in your matrix. So when this happens, this releases your electrons along with H plus. So this electrons and your H plus is being captured in the inner mitochondrial membrane. But this breakdown is done by a first complex that is known as NADH dehydrogenase. Okay, so now your electrons and your H plus are released. Your electrons move into the second complex, but H plus lands here. Ma'am, how here we have only 2 H plus? So at the end, say you have so and so H plus, right? So if NADH plus H plus is converted into NADP, you have plus 2 H plus, right? So it conjugates with FMN and then this conjugates with FES. So here also you have a removal of H plus. So together it forms 4 H plus. So then the electrons move into the cytochrome complex, cytochrome BC complex. This is complex 3. Now the energy from FADH also has to be broken. So that is with the help of complex 2, that is succinate dehydrogenase. This also releases 4H plus. But once again the electrons move into the cytochrome complex. So all the electrons from cytochrome 3, they move into the cytochrome A3. So this is cytochrome BC, this is AC. So when the electrons come here, so it reacts with half a molecule of oxygen and it forms water molecule, one of the product of aerobic respiration. Now since the concentration of the H plus is getting accumulated here, this has to be broken with the help of F0 and F1 ATP synthesis, which in turn gives you ATP molecules. Okay, so for every 2 H plus you have the production of 1 ATP molecule. So at the end of your aerobic respiration, you have to have ATP and your oxygen. So this is ATP from your NADH and FADH alone, right? Yes. So these are the complexes that is present. So complex 1 is FMN NADH. So NADH2 is converted into NAD plus H plus, right? So this is carried by your FMN. The next complex is your succinate dehydrogenase or your ubiquitin or your FADH2. So this is converted into FAD plus 2H plus, because we need the formation of a proton gradient. Then you have complex 3 that is containing a series of electrons. So this is cytochrome Bc, and then you have another complex that is cytochrome A3, and finally it is F0 and F1, right? Yeah. So with this we are done with your ETC as well. So now what is this amphibolic pathway? So your body, in your body you have metabolism. So metabolism is both catabolic as well as anabolic. So catabolic is the breakdown of larger substances into smaller one. So this is catabolic. So here you have the release of energy. So when smaller molecules are used to produce bigger molecules, energy is used up. So this is your anabolic process, this is your catabolic process. Fine? Yes. So fatty acids are broken down to acetyl-CoA before entering the respiratory pathway. So let me tell you something very easy to remember. So this is your amphibolic pathway. So when we have your aerobic respiration, we always think it starts from glucose. It need not be. Suppose if the plant, it's a rainy day, the plant is not able to photosynthesize, it does not have any stored glucose, then how it will produce energy? If suppose the plant has fats, the fats will be broken down into fatty acids and glycerol. The fatty acid will be converted into acetyl-CoA, then it will enter the Krebs cycle. Okay, so if it's carbohydrates, then it forms the normal pathway. If at all, if it's proteins, the plants have only protein stored, then how it will produce energy? So the proteins will be converted into amino acid. The amino acid will be broken down into pyruvic acid, which enters the link reaction, TCA cycle, and the electron transport chain. So everything is interconnected. Fine. So what is respiratory quotient? So during aerobic respiration, carbon dioxide is consumed and oxygen is released, right? So during respiration, the oxygen that is produced as a byproduct in photosynthesis is being utilized and carbon dioxide is being released, right? So the ratio of volume of carbon dioxide evolved to the volume of oxygen consumed is your respiratory quotient. Suppose if I have a reaction like this, so this is the normal equation of your photosynthesis. So C6H12, respiration, right? So you have 6 oxygen molecule, carbon dioxide, water and energy. So how do you find the respiratory quotient? The molecules of carbon dioxide evolved divided by the molecules of oxygen consumed. So it is 6 by 6, that is 1. For all your carbohydrates, the respiratory quotient is exactly equal to 1, which means it is completely oxidized. It is completely combustible. Okay. So in the next case for fats, so tripalmitin is a fat. If my fat is being broken down, so this is the release of energy. So the respiratory quotient is like this. So your respiratory quotient for fat is 0.7. You don't have to remember the equation. You will remember the respiratory quotient for your carbohydrate is 1. So for your for your fats it is 0.7. It is less than 1, right? Yeah. So moving forward. So if it is your anaerobic respiration, so this is anaerobic respiration. So anaerobic respiration has no oxygen. So when it is anaerobic respiration, RQ is infinity. So here when I use an organic acid like this, so here it is greater than 1. So respiratory quotient is equal to 1 for carbon dioxide. It is less than 1 for your fats. It is greater than 1 for your organic acids. And it is infinity for your anaerobic reaction. So all this is what you should remember. Clear? Isn't this chapter easy? So all that you have to remember is your glycolysis, Krebs cycle, link reaction and your ETS. Right? So did I give you a recap? Yes? Clear? Any further doubt? Yes? Any doubts which you want me to clarify in this chapter once again? So tomorrow we are discussing the questions of 2 chapters. One is strategies for enhanced food production. The other one is microbes in human welfare. Right? So we will have 20 questions from each chapter. Ma'am, chemi-osmotic hypothesis, can you explain again? Okay. So chemi-osmotic hypothesis comes in your photosynthesis. So in your photosynthesis also ATP molecule is produced, right? So how is this ATP molecules produced? So a product of your light reaction is ATP, right? So how is this ATP molecules produced? So this ATP molecule is produced because of a H plus gradient once again. So here you have the thylakoid membrane. So the center portion of the thylakoid membrane is lumen. So this is the thylakoid membrane and the outer portion is stroma. So here you have high H plus concentration and here you have low H plus concentration. So this concentration gradient breaks. So the H plus from here tries to come out. So when it tries to come out, your ATP molecule is being formed. So this is your chemiosmotic hypothesis. Okay, did you understand? Clear? Yes, any more doubts? Okay, so chalo, so thank you so much for making the lecture very interesting. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So thank you so much. So this is me wishing you a lot of happiness and lot of good health, wishing you more and more. So stay tuned until tomorrow evening 5 p.m. where we are going to discuss questions from strategies for enhanced food production and microbes in human welfare. Yes, so thank you and hope you have a very good preparation time. So stay tuned and bye.